So we'll start off our genetics lesson by talking about the scientist who discovered it. Gregory Mendel was an Australian monk who studied math and science at the University of Vienna. And he worked with garden pea plants back in the 1860s. And it's with the garden pea plants that he discovered his various laws of genetics. When he began his work, most acknowledged that both sexes contributed equally to an individual. However, they were unable to account for the variations that were seen in family members throughout a generation. For example, they were confused as to why one parent might have red hair, but that his children and grandchildren did not have red hair, and then it might randomly show up in his great-grandchildren. So Mendel started off his genetic experiments using the garden pea plant, which has the scientific name Pisum sativa. He used these because they're easy to cultivate and has a short generation time period. So he was able to study a lot of plants over a short period of time, and so therefore get a lot of data to help him study genetics and then eventually come up with his theories. He also chose this because they normally self-pollinate, but they can be cross-pollinated by hand. He chose true breeding varieties, so that the offspring were going to be like just like the parents and each other. He kept careful records of his large number of experiments. As you imagine, I mean, that would be a lot of information to track. His understanding of mathematical laws of probability helped interpret his results. As we'll see how math is really essential in studying genetics, and later on in this lecture. And then he discovered the particulate theory of inheritance, which just says that that people's physical traits and the variances that we can see throughout a family's generation are all due to minute particles, which they didn't know at this time, but are genes. So here's the general anatomy of one of the pea plants that Gregory Mendel would have used. In the center, we have both the male and the female parts. The female parts include the ovary, which contain the flower eggs, and then the stigma, and then the anther, which contains the male sperm, which is called pollen. So for his experiments, Gregory Mendel cut away the anthers so that the flower couldn't pollinate itself. He then chose pea pods with the specific genes he wanted to study, took the anthers from that pea pod, and fertilized the other pea pod. He used this method to make sure he could control the flow of genes from each generation. So he could control which flowers mated with whom. So these were the 7 characteristics that Gregory Mendel was studying: stem length, pod shape, seed color, flower position, flower color, and then pod color. Throughout his experiments, he found out which genes were dominant and which ones were recessive. Mendel only studied one trait at a time. The first trait he decided to study were the green and the yellow pea pods. Mendel gave each of the generations a different name, like P for parent generation, F1 for the second generation, and then F2 for the third generation. In the P generation, Mendel started off by crossing one pea pod with a yellow pea pod. The F1 generation had only green pea pods, so Mendel wondered what happened to that yellow pea pod. Mendel hypothesized that each of the offspring were carrying the yellow gene, just not showing it. He hypothesized this because he thought the parent alleles separate when their gametes are formed. To see if the yellow gene was in the offspring, Mendel performed a test cross with the F1 generation. And a test cross is used to determine whether or not an individual with the dominant phenotype is heterozygous or homozygous dominant. He allowed the F1 generation to self-pollinate. So here's a picture of the test cross that Gregory Mendel performed. We have our F1 pea plots, so they're both going to be green, and both of them are going to be heterozygous for the pea pod color, which means that they're going to have both the dominant and the recessive genes. The dominant gene is the one that's going to be expressed in the physical characteristics, and in this circumstance, green is the dominant color, while yellow is the recessive color. So we have two heterozygous pea pods mating with each other, and when he did his test cross, he found out that he ended up with 3 green pea pods and then 1 yellow pea pod. And so he discovered that that yellow gene didn't actually disappear. It was just hiding behind that dominant green color. One trait test cross showed patterns. The pea generation showed that if a parent with a dominant phenotype had 2 dominant alleles, then all the offspring would have the dominant phenotype. The F1 generation showed that when 2 heterozygous individuals mated with each other, then the phenotype ratio would be 3 dominant to 1 recessive, and a 1 to 1 genotype ratio. A scientist in the early 1900s took Mendel's ratios and came up with the Punnett square, which helps us to see all the possible offspring that 2 parents could have. To make a Punnett square, you put the possible gametes of the mother and the father on the left-hand side and on the top side of the square. Then you combine the alleles accordingly to see the possible offspring. When the F1 generation were pollinated with each other, when they fertilized each other, the Punnett square showed that the offspring had a 75% chance of having a green pea pod and a 25% chance of having the yellow pea pod, as you can see in this bottom picture here. Mendel took the results from his test cross and theorized that plants carried 2 generations for each trait, and further theorized that gametes only carry 1 trait or 1 gene, and the gametes being egg and sperm, at least in humans, and then in plants it would be the eggs and the pollen. That way, when the gametes come together during fertilization, then the embryo will carry 2 genes for each trait. Because Mendel was a mathematician and a scientist, he hypothesized that the meeting of 1 sperm and 1 egg was entirely random. So you may not know exactly what traits the offspring will have. The Punnett square can only give you an estimated guess of the traits that your children will have. Mendel's work with the pea plants allowed him to come up with his first law of genetics, which is the law of segregation. And this is the cornerstone of his particular theory of inheritance, those particulars being the genes themselves. And the law of segregation states the following. Each individual has 2 alleles for each trait. The alleles separate during the formation of the gametes. Each gamete contains only 1 allele from the pair of alleles for that trait, and then fertilization gives each offspring 2 alleles for the trait. Since Mendel, scientists have discovered that each gene has a specific place on each chromosome, and this is called the gene locus. This allowed scientists to come up with a chromosomal theory of inheritance, which states that offspring will inherit half of the chromosomes from 1 parent, with each chromosome carrying a bunch of individual genes. Upon fertilization, the new offspring will have a full set of genes from both parents. So I know I tossed around a couple of really big vocabulary words in those first couple of slides, so let's break those down a little bit more. So an allele is going to be an alternative form of a gene. Dominant alleles will mask the expression of the recessive allele, and for the most part, an individual's traits are going to be determined by the alleles that they inherit from their parents. And alleles occur on homologous chromosomes at a particular location called the gene locus. So each person contains 46 homologous chromosomes. You're going to receive 23 from your parent, from one parent, and then 23 from your other parent. Each pair of homologous chromosomes are shaped the exact same way and carry the exact same genes. So here we have one pair of homologous chromosomes. Notice how they're the exact same length and width, and then they carry the exact same number of genes. So we have the G gene on top, the R gene second, we have the S gene third, and then we have the T gene next. So the T gene is going to be located on the exact same place on one chromosome as it is on the other homologous chromosome, and this is called the gene locus. And for this particular pair of alleles, it is going to be heterozygous because we have one recessive allele and then one dominant allele. And it looks like for all of our for all of our gene pairs, all of them are going to be heterozygous. So the only exception to the homologous pair rule is going to be the sex chromosomes. And so we have the X and the Y gene. The X chromosome is going to be a lot bigger than the Y chromosome. And because it's a lot bigger, it's going to carry a lot more genes. And the Y chromosome is going to be a lot smaller, and so therefore carries a lot less genes. Even though that they both determine the sex of a baby, they are not homologous since they are shaped differently and carry a different number of genes. So the genotype for a person is the alleles that the individual receives at fertilization. A homologous genotype is when there are going to be two identical alleles, and there can be homozygous dominant when a person has two dominant alleles, or homozygous recessive when a person has two recessive alleles. And then heterozygous just means that that person has two different alleles for the same trait. And then the phenotype is going to be the physical appearance that is determined by that genotype. And then again, there is the phenotype is not entirely determined by the genes, as the environment has some influence over the physical characteristics of a person. Normal skin pigmentation for skin is a dominant allele, and by that I just mean that the person is able to produce the particular protein that produces melanin for the skin, which is what makes your skin turn darker after you get a sunburn. If a person carries two of these dominant alleles, then the person would have normal skin pigmentation. If the person carried one dominant allele and one recessive allele for albinism, then they would still have normal skin pigmentation. If, however, the person received two recessive alleles, then they would end up having albinism, since they wouldn't be able to produce melanin and would lack normal skin pigmentation. Mendel wanted to see if two separate traits were inherited together or separately. And to do this, he got plants with all recessive traits. And in this case, he got the recessive traits he chose to study were short plants and yellow pods. And then he mated them with plants with all dominant traits, which the dominant traits he chose to study were tall plants with green pods. And just like in his first experiment, these were true breeding, which just meant that all of the plants that had the dominant traits would all produce offspring that had plants with the dominant traits. Same with the plants with the recessive traits. And also, like in his first experiment, all the offspring in the F1 generation were heterozygous. Mendel hypothesized that if the alleles were inherited together, like all of the dominant traits sticking together and all the recessive traits sticking together, there would only be two phenotypes that the F2 generation would have: tall plants with green pea pods and short plants with yellow pods. If the alleles were inherited separately, then four phenotypes would be possible. And the offspring did show these 4 different phenotypes. Using a 2-trait Punnett square, it shows the likelihood of the offspring inheriting each phenotype. Because each plant were heterozygous for each trait, they were able to produce 4 types of gametes. As with the 1-trait Punnett square, the mother's gametes go on one side and the father's gametes go on the other. 9 of the offspring had both dominant traits. 3 had the dominant trait for height, but the recessive trait for seed color. 3 had the recessive trait for height and the dominant trait for seed color, while 1 had both recessive traits. Out of the 16 offspring, there is 10 different genotypes and 4 different phenotypes. This led to Mendel's second law of genetics, that genes separate independently of each other while the gametes are formed, and that each gamete can have any assortment of these alleles. Anytime 2 organisms with 2 heterozygous traits mate with each other, the phenotypic ratio will always be 9 to 3 to 3 to 1. 9 of the organisms will have both dominant traits. 3 will have the dominant first trait and then the recessive second trait. 3 will have the recessive first trait and the dominant second trait, and then 1 will have both recessive traits. So fruit flies are commonly used in genetic research today for the very same reason that Gregory Mendel used pea pod plants in his genetic research, because they reproduce very often and have short lifespans. So the scientists are able to get a lot of data really quickly. Here's an example of a 2-trait test cross using fruit flies. And the traits that we're going to study are the color of the fruit fly and then the length of the wing. So long wings are dominant over short wings, and then gray bodies are going to be dominant over black bodies. So in our pea generation, we know the phenotype of our flies, but we don't necessarily know the genotype. We know that the phenotype of our pea generation is going to be dominant. So he has long wings with a gray body. In order to figure out the genotype of our fly with the dominant traits, we're going to need to choose a mate that we definitely know their genotype. And we can know the genotype of a fly that has all recessive traits. And so in this case, we're going to mate our gray-bodied, long-winged fly with a fly that has short wings and a black body. And after mating our flies, we see that some of the offspring had both recessive traits. This tells us that our original fruit fly was heterozygous for each trait. This Punnett square shows us all the possible genotypes of our offspring. The eggs from the fly with the recessive traits only has eggs with the recessive traits. And the other fly's gametes has 4 possible gametes because it's heterozygous for both traits. With this cross, the potential offspring will have 4 potential phenotypes and genotypes, but in a 1 to 1 to 1 ratio. So the Punnett squares that we just saw assumes both of Mendel's law of inheritance and that male and female gametes combined at random. Punnett squares can get really complicated, but thankfully there's an easier way of figuring out the probability of the offspring inheriting one allele or another. And it is called the rule of multiplication. You will still need to perform a Punnett square for each individual trait, but to figure out the probability that the offspring will inherit a certain combination of alleles, all you have to do is multiply the probabilities together. For example, when you flip a coin, the odds of getting tails is 1 to 2, but the odds of getting tails when you flip 2 coins is going to be 1 over 2 times 1 over 2, which equals 1 over 4. So the probability of getting tails twice in a row is going to be 1 out of 4. We can apply Mendel's laws to human genetics, especially to see the probability of our kids inheriting a certain genetic disorder. If you are worried about your kids having a certain genetic disorder, you can talk to a counselor who will use a pedigree to determine the probability of your kid having that disorder. And a pedigree is the chart of a family's history in regards to a certain genetic trait. Males are going to be characterized by squares. Females are characterized by circles. Shading represents individuals with the genetic disorder. Horizontal lines between circle and square is a union, and then a vertical line down represents children of that union. Autosomal disorders are found on any one of the 44 chromosomes, except the 2 sex chromosomes. Recessive disorders, like recessive alleles, only affect people if they have 2 recessive alleles. As long as a person has 1 dominant allele, then the person will not express the recessive disorder. However, if they do have children with someone who is also a carrier for the recessive disorder, then their children could potentially have it. Both parents should get tested to see if they are carriers for a particular recessive disorder. So with recessive disorders, if one of the parents has the disorder, then the recessive gene will be passed on to the children. The children will then be carriers for the disorder, but not necessarily have the disease. In our pedigree here, we have one grandmother with the disease, shown by this shaded circle, and she has 2 daughters, and each will be a carrier for the affected gene. The daughters may pass down the affected gene to their children. And if their mate also carries the affected gene, then their children could end up with the disorder. This is why certain diseases skip 1, 2, or even 3 generations. In this particular pedigree, our 3rd generation has 2 cousins mating, and both of their children end up with the disorder. And this is why children of close relatives this is why there are a lot of genetic abnormalities in the children of people who are closely related to each other. So disorders caused by dominant genes are going to be a lot easier to detect, since the dominant gene will show up in the phenotype. If both parents do not have the disorder, then the kids definitely will not either. However, if the parent does have the disorder and is heterozygous, it's possible that the children will get it too, or the child may not. So in this particular pedigree, we have 2 heterozygous parents with the disorder, and they have 1 boy and 3 girls. The 1 boy and 1 girl has the disorder, while the other 2 girls do not. The girls without the disorder that mate with unaffected males will have children without the disorder, while the boy and the girl with the disorder may or may not have children. May or may not have children with the disorder, since they may or may not pass down that dominant gene. So cystic fibrosis is an example of an autosomal recessive disorder. This is the most common lethal genetic disorder among Caucasians in the United States. And what cystic fibrosis does is shut down the chloride ion channel located in mucous membranes, and this causes abnormally thick mucus. The thickening mucus can be in the lungs and pancreas. It causes affected people to have a really hard time breathing and a hard time digesting some foods. And Huntington's disease is an example of an autosomal dominant disorder. The person has a mutated Huntington protein, which causes a progressive degeneration of neurons in the brain. The top picture shows the number of neurons a healthy person has, while the bottom picture shows the limited amount of neurons a person with the disease has. Unfortunately, symptoms don't appear until later in life, so usually after people have children, which means that they could have passed on the gene without knowing it. With genetic testing today, though, people can get tested before having children to see the likelihood of them passing on that gene. So now we'll move on to section 3 and continue to discover the complexity of genetics. So incomplete dominance is when both alleles for a trait is not dominant. So here, for example, if a snapdragon flower has 2 different genes for its petal color, one red and one white. If the snapdragon flower inherits 2 red alleles, then it will have red petals. If it inherits 2 white alleles, then it will have white petals. If, however, it is a heterozygote for this trait, then it will have pink petals. And so we have two alleles here being equally expressed in the phenotype. An example of incomplete dominance in human is wavy hair. So we have one gene that could give people straight hair, one gene that can give people really curly hair, and if a person inherits both of these alleles, then the person will have wavy hair. Since Mendel's time, scientists have discovered that many traits actually have more than 2 possible alleles, and these are called multiple allele traits. Blood type is an example of this, as there are 3 possible alleles, A, B, and O. The A and B allele are dominant, and the O allele is recessive. If a person inherits only the A or B allele, then they will have type A or B blood, respectively. If they inherit both the A and B allele, then they will have type AB blood. If, though, they inherit both recessive alleles, then they will have type O blood. Type AB blood is also an example of codominance, which is when both dominant alleles are equally expressed in the phenotype. With codominance, when 2 heterozygotes for A and B mate, their offspring will show a 1:1:1:1 phenotypic ratio. The children have a 25% chance of getting AB blood, type A blood, type B blood, or type O blood. When a physical trait is determined by 2 or more genes, or 2 or more sets of alleles, it is called polygenetic inheritance. Each dominant allele the person inherits will have an additive effect on their phenotype. For example, height is controlled by many genes, and the tall allele is going to be the dominant allele. The more dominant genes a person has, the taller the person will be. Polygenetic traits are generally expressed by a bell-shaped curve. Genetics and the environment come together through multifactorial traits. Some genes are activated when they come in contact with a person's environment. Like the sun will turn on the protein that produces melanin in your skin. So the more you are out in the sun, the more melanin you'll produce, and the darker you will be. Some other things that could happen as a result of a person's environment is diabetes by the person eating too much sugar, schizophrenia by a person undergoing too much stress, allergies, or even cancer. So here is one of our polygenetic traits, height, represented by a bell-shaped curve. As many as 700 alleles will affect someone's height. The more dominant alleles a person has, the taller a person will be, while the more recessive alleles the person has, the shorter the person will be. And a bell-shaped curve, this bell-shaped curve shows us that very few individuals will be really short and very few individuals will be very tall. Most individuals are going to have an average height somewhere, or they're going to have a very even amount of recessive and dominant alleles. Skin color follows the same pattern as height. Skin color can be further varied by the sun, as the sun causes the skin to produce more melanin and therefore more pigment. If a person moves to a tropical area, the long exposure to the sun can cause a permanent darkening of the skin. One's eye color is a result of multiple genes interacting with each other. If a person has the dominant gene, they will have brown eyes. If a person has 2 recessive genes, then they will have blue eyes. The person may also have blue eyes, though, if they have 2 copies of one other gene. These 2 genes will override the eye color gene. And if the person isn't able to produce any pigment in the eye, as with albinism, then they will have red eyes, as shown in the left picture. So most of the time, a single set of alleles will only have one effect on a person's physical traits, but sometimes they will have more than one effect, and this is called pleiotropy. This can be really seen in a particular disease called Marfan syndrome, and this is due to a single pair of alleles producing an abnormal connective tissue, and this single set of alleles is able to affect so many parts of the body because connective tissue is found in many parts of the body. With Marfan syndrome, there will be a mutated FBN1 gene, which is located on chromosome 15. And due to the poor connective tissue, they will have long arms and legs, poor eyesight, and a weakened aorta, which is a part of the heart. And this is because the FBN1 gene codes for connective tissue, which is really important in the supporting of the ligaments, hearts, and eyes. Although one gene may not affect the heritability of another, if 2 genes are found on the same chromosome, then they will be inherited together. This is because a gene is found on the same chromosome in all humans. An example of linked genes is the body color and wing size in fruit flies. If a fruit fly is heterozygous for both traits, then they can only produce 2 kinds of gametes. The specific alleles for body color and wing size found on one chromosome will be inherited together. As discussed earlier, females have 2 X chromosomes and males have 1 X and 1 Y chromosome. All of the eggs of the female will have the X chromosome, and the sperm for males could either have the X or the Y chromosome. The sex of a baby and some genes go together. When a gene is found on the X chromosome, this is called an X-linked gene. If it is found on the Y chromosome, then it is called a Y-linked gene. It is the SRY gene on the Y chromosome that gives males their primary and their secondary sex characteristics. And so this would be an example of a Y-linked gene. Sex-linked traits can be confusing. For example, the gene for vision is found on the X chromosome. When studying this trait in fruit flies, the P generation had the female with normal vision and the male with color blindness. After mating the F1 generation, the F2 generation showed 3 flies with normal vision and 1 fly with color blindness. The one with color blindness were all male. This shows several patterns. If a male gets an X chromosome with a recessive disorder like color blindness, then they will have color blindness. If the female ends up with a gene with the recessive disorder, she may or may not have the disorder. And this is because she has another X chromosome that could contain a normal gene for vision. Since this allele is dominant, she will have normal vision, but she will be a carrier for color blindness. Here, a male with normal vision has children with a female with a carrier of color blindness. All of the females will have normal vision because they get this dominant allele from their father. Regardless of the allele they get from their mom, they will have normal vision. The males have a 50% chance of having normal vision or color blindness since they only have one allele governing this phenotype. So X-linked disorders show some patterns. Sons inherit traits from their mother, since the son's X chromosome comes from their mother, More males than females will have the disorder, as the allele on the X chromosome in males will always be expressed. Females who have the condition inherited the mutant allele from their mother, or from both their mother and their father. And then conditions appear to pass from grandfather to grandson. There's very few X-linked dominant disorders, and some of the patterns shown with these disorders is that the daughters of affected males will have the condition. Affected females can pass the condition on to daughters and sons. And if the father is normal, then each child has a 50% chance of inheriting it. There are also only very few Y-linked disorders. And some patterns shown with Y-linked disorders is that they will be present only in males, since only males will have the Y chromosome. So this here is my cat Carmel, and she is a tortoise-shelled cat. And tortoise-shelled cats are walking genetic lessons. The genes for fur color in cats is found on the X chromosome. Each female has 2 X chromosomes per cell, but only 1 X chromosome can be active at any one time. The other one shrivels up into something called a bar body. The gene for fur color on the active X chromosome will show on the hair that is growing from that cell. Tortoise-shelled cats are heterozygous for fur color, meaning that they will have both the gene that makes the fur orange and the allele that makes the fur black. Therefore, some of the cells will grow orange fur, and then some of the cells will grow the black fur. And this is what gives tortoiseshells their bicolored coat. And this is the reason tortoiseshell cats can only be female. Males only have 1 X chromosome per cell, and so whatever gene is on the X chromosome will show on the fur. Males will either be all black or all orange. The value of any one allele is going to be determined by the environment that that allele is found in. And so that's why it's really important to for the recessive and dominant alleles to be maintained in an animal population so that the genetic diversity can stay high for an animal population. That way, if there was an environmental change that might, say, make a recessive allele more valuable than the dominant allele, then that recessive allele is still within the gene pool, so that those individuals with that recessive allele can reproduce more, and so more of the animals will have that recessive allele, and so would be able to survive longer in the changed habitat. And this is why heterozygotes are so important and have this advantage, since they can protect that recessive allele. So not only do the different genetic laws affect genetic patterns, but so do how animals reproduce. Animals can reproduce either sexually, where there's 2 parents, or asexually. And asexual reproduction can come in several forms. The main one is binary fission, but there is also generation from fragmentation or budding. With asexual reproduction, there is 1 parent, and the offspring will contain all genes from that parent. In other words, it will be identical to the parent. So besides binary fission, budding is also another way that animals can asexually reproduce, as shown with this hydra here. And the offspring is going to be born by budding from is going to be born when a bud is formed on the parent, and then the offspring will eventually mature from this bud. So a lot of animals and plants reproduce through sexual reproduction, and this includes 2 parents. Each parent produces corresponding gametes, eggs or sperm. When the sperm fertilizes the egg, a zygote is formed and will develop into a baby. Some species, like earthworms, are hermaphrodites, which just means that they contain both male and female parts. However, they will still cross-fertilize to mix up the genes of the offspring to help maintain genetic diversity. Parthenogenesis is a type of sexual reproduction, but with a twist. Queen bee eggs can develop into an adult bee, whether fertilized or not. The fertilized eggs are diploid, meaning that they contain 2 genes for the same trait, and become female workers. The unfertilized eggs are haploid, meaning that they just contain one allele for the trait, and become male drones. In some animals, the gonads may change function depending on the environmental conditions. For example, in a coral reef fish called a rhasis, a male has a harem of several females. But if the male dies, the largest female becomes a male. Reproduction in the slipper snail involves a forming of a stack of individuals. The individual at the top of the pile will always be male, and the rest will be female. So fertilization during sexual reproduction can occur either on the outside or on the inside of the animal. Many aquatic animals do external fertilization. And this is when the egg and sperm are released into the water, and the sperm then fertilizes the egg. All land mammals do internal fertilization through copulation. Some aquatic animals, like lobsters and crayfish, also undergo copulation. So after fertilization and the baby is fully developed, there are a couple of different ways for the baby to come out in the world. It could be hatched from an egg, or the egg could hatch inside the mother, or the mother could give live birth. Oviparous animals will lay shelled eggs, and some animals that will do this are birds, fish, and reptiles. In shelled eggs, there are extra embryonic membranes to serve the needs of the embryo and prevent drying out. Ovoviparous animals actually have the eggs inside of their body until they hatch, and then they will give birth to live young. Some example of animals that do this are oysters, seahorses, and garter snakes. The young from the ovoviparous animals are generally capable of feeding on their own. Animals that give birth to live young are called viviparous. And mammals do not lay eggs because they have the placenta to nourish and keep the embryo hydrated. Some mammals, though, do lay eggs, like the spiny anteater and duck-billed platypus. Marsupial mammals do give birth to live young, but in an immature form. These babies will need to crawl to the mother's pouch and attach to a nipple where they will continue to develop.